Okay. Well, okay. Welcome to the August 24th lecture, the first lecture of class. And hopefully everybody can hear sound well, which is good. And what we want to share is my computer screen, because what I want to do is to sort of come into, if I can find the right one. That's not what well, let me see. Pull that back, see if I can go backwards on this one. Yep, okay. Now, Russell, are you seeing everything that's on the screen?
Yep.
Okay, because what will happen here is that my computer will project on the screen. Everything that Russell can see in his little computer is what the students can see, and it also should be archived because it's shared. Okay, so that's why Russell's there, to make sure when I pull something up it's visible on his screen. That's the test for the archival parses. Okay, so what we wanted to do is, we're in our self-pollinated crops course. Now we have a sort of a bond between those who are taking it resident and those that are taking it distant. Okay, the resident students will take the class. And Tom, you might want to open that door in case there's a late student that comes in, so they can see that we're actually here. If it gets too noisy, we'll close it. But I'm thinking there should be one more student coming that I'm aware of. 2, 4, 5, 6, 7, 8, 9. Could be 2 more taking it. Should be coming. Anyway, so what we have is the resident students. When you have something which doesn't make sense, not to you, because I'm sure everything makes perfect sense to the resident students, but when you think your neighbor probably didn't get it, or maybe one of the distant students didn't get it, you can ask a question, and you can ask a question at any time. We'll never assume how different or difficult or easy the question is. You're asking it for yourself. We know you're just being really polite and asking it for your neighbor or for a distant student, okay? So you should never be embarrassed about any question that you ask. So that's the goal of the resident class. For any distant student who is taking the class, your goal is to make sure that when I'm talking, A, you can hear me, so we didn't kill the sound by doing something stupid, and B, you can see the screen, because when I archive it, there's nothing worse than me archiving either a lecture for which no sound was recorded or no screen was actually put up. Okay? Thanks, Dudema. Okay? Now, we'll always start at 4 o'clock, but again, like I say, whether you take the class synchronously or asynchronously is entirely up to you. I never take attendance. Okay? Because I know that you'll be taking it online at a time that's convenient for you if you have a conflict. Okay, a couple of things that we want to look around in. First of all, the announcements. The announcements, this is how I'll communicate mainly with you. So anything that comes up, like if there's a difficulty at the bookstore, you can't buy your notes, we'll need to find out how many more students need to buy notes, and then we can put an order in to make that number. Okay? In the interim, you can download the PDF files, which are there. Okay. We're in Plant Science 272, and if you're worried about your genetics, you know, like you've been out of school for a little while, you're coming back, and now you're worried that you may not know your genetics, I highly recommend the Plant and Soil Crop Technology Library. It's usually called the E-Library. It's got a lot of great information on refreshing your genetics, biotechnology, things like that. So it's really good on that. And one thing that you need to do is the online classroom and the lecture archives. The online classroom is always for the synchronous lecture. If it's not between 4 o'clock and 5:20 when we have the class, that's the only time on Tuesdays and Thursdays. That's the only time the online classroom is open. Every lecture, hopefully, within 5 minutes of the end of the class, I have archived, and it's in the lecture archives. So let's assume, and you know, truly life does happen. Let's assume that you need to go to a doctor's appointment or you have to see a professor at 5 o'clock. So you need to leave the class at 4:30, no problem. You go out, you get your business taken care of, you go online. In that case, because the class is no longer synchronous, you go to the lecture archives. Okay? So, and most of these I will be sending you also by email. Almost every one of these announcements, there's a where I can click on it and send it by email. So how many of you received an email already saying that we migrated the sections and that everybody, whether you came in as 951 or as 700, received the email? Everybody get one? Okay. No. Okay. Okay. Okay, Roberto, what I want you to do is check your email in your student ID and make sure that it's the email you're using. Okay? Sometimes, like if you came here and you started out with a personal account of Hotmail and then you switched to Gmail, the university remembers you only as your Hotmail account until you change it, and so it gets sent to hyperspace. Okay? So that's something, this email system only works if the email you're using is actually the current one. And because many of us have been here for a couple of years, you know, you can change emails, or it could be one that you're not using or looking at very often. Okay? So that's something you have to watch. Now, so that's how I'll do it. That's how I'll do the announcements. One thing we'll do is we'll go to the discussion board, and we can do that next. Now, again, help me out on this one. I'm hoping this will come right up and share. It's a little slow sometimes, but it should be up there. Okay, so it came up. Good. Now, Russell, help me. Can you see on the side in case any student is asking any question?
Yeah, I can see.
Okay, great. Okay. See, I can't see that on mine. Mine's full screen. Okay, class introductions. It's always nice to know who your classmates are. So it would be great if you could come in here and just give a little information about yourself. That way we'll know who you are. And usually we get some pretty amazing people. I mean, it's kind of fun. You'll be surprised. We have people all over the world. Some are all over the world in the classroom, because we're from all over the world here. And some are from all over the world actually in situ, back where they are. Okay? We have a lot of commercial people who take the course for professional development, and because of that, you get, you'll get to see some interesting comments from people very much plant breeders in a very practical world. And you'll see them, like one of the students I know has already got to go to Brazil for 2 weeks in September. So just, things like that, they'll get different approaches. So you do want to look at that. If you have any questions on the course materials, this is where I'd like you to ask them. The reason for that is very simple. If you have a question, trust me, one of your colleagues will have a question or a very similar question. And so the best way to ask it is to ask it on the discussion board. Then when I answer it, everybody can see the answer. I'm not sure if I've allowed anonymous posts, but if you need an anonymous post that makes you feel more comfortable, that's fine too. But remember, we always assume that you're asking the question out of politeness for somebody else that's just too shy to ask the question, and you're just asking it because you know that they would ask it. You knew the answer clearly, but you just thought your friend didn't know the answer. Okay, so that's fine. We also then will go into our assignments, and I don't want you to start on these quite yet because I'm not sure that we've got these all updated completely. But the problem sets, there are 3 problem sets. Now, these are suggested dates to submit them. I don't go by a strict date. All I go by is at the end of the first module, you should be done. Okay, and even that we're flexible on. I think it's best if you try to stay current, and the reason for that is that if usually when you hear a lecture, everything makes perfect sense, right? The professor is brilliant, you're brilliant, everything works perfectly, right? It's then when you do your first problem, you discover that the professor was an idiot. You're still brilliant, but the professor didn't explain it in a way you can handle, right? So the short answer is you want to be doing those problem sets to determine whether or not the professor has really got anything to him or not, right? So you want to do that current so you don't get, so far behind nothing makes sense. Okay? The threaded discussions are usually due at the last, like the Tuesday before the end of the first module. There's a couple of benefits by answering them not too quickly, but soon. There's a waiting, if you have the benefit of another person's answer, clearly your answer should be better, right? So the bar gets raised every time somebody answers, okay? So unless you have a really neat way of presenting something, if you're the 8th person in there, you might want to go to a different question where you can be the second person, okay? That's one thing. Now, the other thing I would say on this is, just do your best, and you only need to answer one. I remember a student thought they had to answer all 7. It was like, whoa, no, you don't have to do that. And it makes a real problem for me grading because I don't know which one they want me to grade. Clearly, I could grade all 7, whichever you get the highest score on, that's the one you'd want, right? Okay. Now, the short paper, and I want you to be shameless on this paper, okay? It's for it's for the academic for the course. Okay, now I should mention, we have students who are taking this for credit, for graduate credit. They have to fulfill all the assignments. There are other students who are taking it for non-credit but would like a certificate. They don't need to write the paper, okay? They really don't need to do the threaded discussion, but often they can add a lot of really nice stuff for the threaded discussion. But what I would like to do if you want a certificate is to do the problem sets and the final exam, because that way I will know whether or not you've mastered the subject matter. I don't want you to pay, $135? Never come to the class and say I'd like a certificate that I'm a bona fide plant breeder at the end. So we have to have some credible way of knowing that you actually understood something. So for those of you that are non-credit, and I think everybody in the resident class is for credit, you will take all the assignments, but for the non-credit students that want a certificate, they need to do the problem sets and the hour exam. And if you don't want a certificate, you can sit back, kick back, and just use the lectures to put you to sleep on a late night story or something. Okay. So we're very flexible for everybody. Okay. Now on the short paper, what I say about being totally shameless is it should be brief. It should be 2 pages, single spaced, 4 pages double spaced. Okay. It'll be 20% of your grade. You pick any self-pollinated crop you're interested in. Anyone, okay? And from the literature, trace the origin of the crop, describe its uses, its relative contributions to humankind, and how it is bred. Anything significant about the breeding methods and production practice that have contributed to improved performance, okay? Now, for your sake, do each one of those. Okay, where students often get confused is they do a great job of discussing, say, the origin of the crop, and they never talk about breeding. Well, what's the title of this class? Breeding for self-pollinated crops, right? So you ought to have something about breeding. Maybe they do a great job on breeding, but they never talk about how it's used. So you can't tell if alfalfa is eaten by cows or consumed by humans. That's not very good, right? So you want to cover its origin. You want to cover its uses, its contributions to humankind, and its significant breeding methods and production practices. Okay? You cover all those, you'll be fine. Now, sometimes there's some confusion as to what scholarship is. Scholarship is writing in your own words. Occasionally I've had a paper turned in where if I Google the sentence, it takes me right to Wikipedia. That's not scholarship. Okay, that's copying. So you want to write in your own words. Okay, if you want to quote something, someone said it particularly well, then make sure you put it in quotes. Make sure you put in the citation where you got it from. But don't make your 2-page paper a 2-page quotation. Okay, it's got to be your work, and it's got to show that you've thought about it. Now, when I say be totally shameless, how many of you are working on wheat as a master's or PhD program? Okay, 2. Okay, 3. How many of you are working on soybeans? 2. Okay? How many of you are working on, say, well, what's another good self-pollinated crop? Barley or any other self-pollinated crop. How many people? Sorghum, okay. Now, the sorghum's interesting because sorghum, I don't want you to do this, Roberta, but you can actually write it as a self or a cross-pollinated crop because it's sort of half and half. So whichever one if you're going to take the cross-pollinated crop class, if you use the paper for the self-pollinated crop, I don't want to see it for the cross-pollinated crop. Okay. Okay. Pardon me? Proso millet. Is that self-pollinated? I don't know. Okay, I'm thinking millets often are, but they could be cross-pollinated. Actually, I think it is self-pollinated in that case, okay? Write about the crop you're going to do your master's or PhD on. I mean, what's a better prelim question than to ask somebody, tell me where wheat originated? Or where did the 10 accessions that built the American soybean industry come from, okay? Be shameless. Use this paper to get you started on your literature review for whatever degree you're working on. Okay? And if it's not, do it for amusement. You know, like you've always wanted to read about asparagus because it's cross-pollinated. Well, when we get to the cross-pollinated crop module, work on asparagus. Okay? You can do that. So it should be of interest to you and it should have some benefit. So I would definitely do this for, when you're working on, on a crop that you're interested in working on. Okay? That makes sense? So that's how we're going to grade. And by the way, all the problem sets are electronic and the hour exam is electronic. And because everybody will take it at different times, don't put on the discussion board, I answered question 6 and I disagree with you. I think it should be B instead of A because you've just given the answer to all your colleagues. What you want to do is when you click on it, you can say show results and it will give you my reasoning behind it. If you disagree with my reasoning, then you can come see me or email me. But in that case, you want to email me. Don't use the discussion board, okay? Does that make sense? Okay, very good. Okay, now, let's see what else we want to look at. The external links sometimes can be quite useful. Things that you can look at, additional references, some of the graphs we use. It's a useful plant breeding site. It has a pretty good glossary in it. If you're interested in African crops, crop technology, some of the genetics education centers, things like that. If you're looking at genetic resources, things where you get germplasm from, there's some pretty interesting ones that you can find there too. Okay. Now, let's go back. And all this is still on the archives or sharing, right? Yeah, I hope so, man. Okay, what I should do. There's one part that scares the devil out of me. Okay, so is the screen blank now for you too? Okay, we're going to stop sharing, and now we're going to share my computer screen, and we're going to come into I guess we'll look at the chronology of plant breeding first. Okay. I guess we didn't really go through the syllabus at all, did we? Should we look at the syllabus? Would that be of value to you? Maybe we should talk a little bit about that too, because I hadn't thought about that. So we'll go back into, I think it's Blackboard. That did come up, right? Okay, and we want to find the syllabus. That's slowly coming. Sometimes Blackboard gets really slow, which is very annoying. Well, I tell you what, maybe I'll just back out of this thing for a second. Try the syllabus. Okay. Well, we won't worry about the syllabus. This gets a little scary at times. Okay? What we'll do then is we'll come back into here, we'll stop our sharing, and now we're going to share my computer screen, and we'll go into the chronology of important facts. Now, can you see that? Okay. And is it within the window? Okay, see, mine again will look a little funky. I'll make mine full screen here because I'm just very concerned. They changed Blackboard, so this used to be very simple, and sometimes it's not nearly as simple. It used to be this would come up in the window cleanly. Remember that, Tom? And now it appears that it doesn't. It's in the window when you see it distance, but it's not when you're in resident, so you still have trouble on that. Okay, what I really want to do now is just have something up on the screen while we talk about textbooks, okay? The only book that you will need for the class is the Principles and Practices, okay? The way it's set up is color-coded. The first module is gray, the second module is orange or yellow, and the last module is blue, and Tom Hogemeyer, who's in the back, will be the professor that'll teach you the third module for those of you that are lucky enough to be enrolled, okay? The way it's set up is so that there's typing on one page, but only on the front. On the back, it's empty. So the idea is go ahead and write all your notes on the back, okay? Now, one of the questions that we'll have to do, and I'll try to remember to do this tonight, is to ask people how many are missing the course notes. So who does not have the course notes that would like the course notes? 1, 2, 3, 4, 5, 6, 7. Whoa, 7 in here, okay. Okay, that's 7 of the resident students, and the distance students we'll find out from them also. We'll query that. Now, did you check to see if it might be down on city campus at all? You might. Okay.
If they would have it on city campus and the guy.
Okay, okay. Sometimes, okay, first of all, I will always repeat the question. And the question is, did anybody check to see if it was on city campus? And the answer was, at least when you talk to someone in the East Campus, that it was probably not be in the city campus. They may be mistaken there because in order to get the book online, they have to send a few copies to city campus. That's the only way. I mean, it's funny. The bookstore doesn't know that it's actually anywhere until there's a copy down on the city campus bookstore. Okay? Now, you had a question or a comment?
I was there today and they had a hardbound book.
Right.
$100 book.
Right.
For this class.
That's recommended if you're going to be a plant breeder. Okay? And we're going to get to that. The only one I want you to buy is $15. Okay? It's the the copying costs and nothing more. Okay? Tom and I didn't want to have a copyright on this and didn't want to have you pay $20 and split the $5 extra. So that's what it is. I may have to run out and get a drink of water, I think. Excuse me for a second. I'll be right back. I'll get a drink of water. Oh, thanks, Tom. First day this happens. Usually by the end of the semester I can talk forever. And it just seems like that way even after the first 10 minutes. The book that I would recommend, but it's not required. Did they say it was required?
It did say required.
Man, they do that every year. And every year I write in our notes that it's recommended, not required. It's a great book. It's by George Aqua, and it's the Principles of Plant Genetics and Breeding. It's a very modern textbook. It's as good a textbook as I've seen. Thank you, Tom. So it's really excellent. So if you're going to be a plant breeder, sometime in your career you may want to buy that book. Okay? Now, there's lots of other books. The book we used to use was Principles of Cultivar Development by Walt Fair. Okay? It's an excellent book, very good on theory. Okay? I personally like the Aqua book a little better. Okay? But this one is used, and it's very good, and it's not very expensive. It's, I think, something like about $25-30. It was originally published by a major publisher, and then they decided not to republish it, so Walt had it republished, and you buy it from him. I think the Iowa State Bookstore is the one that has it. So that's a good book. Another book, which is pretty good, is Plant Breeding Theory and Practice by Strokoff, Tomes, and Christie. This one is now published in India. Okay? And the beauty of that is, if you're unfamiliar with Indian publishers, is they don't have the international copyright laws. So any book that's out of print that's still in demand, they can publish. So they provide a really useful service. So this book won't be quite the high quality paper that maybe is in the copy I bought, because I bought it new, and I paid about the $100. And it's let's see who it's published by. Westview Press, which is an academic publisher. Well, you'll get it from an Indian publisher, and it'll be probably under $25. Okay? So for example, if you ever wanted the statistical text by Federer, which is one of the classics of all times, and still probably some of the best for incomplete block designs and things, you'll never buy it by a US publisher. You go to India and you get it for $12 because it's used so widely that it's that inexpensive. So this one, you know, by Strohskopf you can get. There are some modern ones. Jensen wrote Plant Breeding Methodology. To me, it's a little bit The World According to Neil. I think Neil is still alive. I knew him when I was a young man, met him. He was a breeder at Cornell at the time. It's a good book, but it's good in some areas, better in some areas than others. It's kind of eclectic. There's a brand new one that came out by Brown and Caligari, Introduction to Plant Breeding, and that's maybe a couple years old. If you know these two guys, Caligari is probably one of the best quantitative geneticists that was of his vintage. It's a pretty theoretical book. I don't find it as valuable as Aqua's book. Aqua's book is more general, more easy to read, that type of thing. Now, the book that I actually learned was Allard, which I didn't bring down. At the same time, Briggs and Knowles wrote a textbook, and when I first started teaching this 25 years ago, this was the textbook we generally used. Okay, it's pretty good for some of the historical work. You know, it was published in there's a funny story. Allard and Briggs and Knowles decided to publish a textbook because they know they needed it, and then the authors as a group couldn't get along. So Briggs and Knowles published theirs. Allard published his. This one came out in '66. Allard's came out in 1960. So you can imagine which one took most of the breeding market. Well, it was Allard's book. Plus, he was brilliant. It's a good book. It's good for historical things, but things like single seed descent were not rediscovered until about 1966, 1967. So there's no single seed descent breeding in this book. And then the last book that I really like is this one by N.W. Simmons. Okay? Now, N.W. Simmons has a book called Principles of Crop Improvement. He's a really good author. He's a banana breeder, okay? So you got to give him credit for that right off the bat, because bananas are clonally propagated. It's a very difficult crop to work with, and that's what Norm worked with. He worked with bananas, okay? And what I like about this book is he's a no-bones plant breeder. So you read an American textbook that's based on corn breeding, and they'll have every whistle and bell you can imagine on recurrent selection. You read Norm's book, and in about 2 pages he says, look, there's 3 things you do. You form families, you evaluate families, and you recombine. You call the program on how you form your families. Half SIB, full SIB, reciprocal, recurrent selection, all that. Has one diagram and says this is the whole story. Everything else is a whistle and a bell. So if you want a very efficient, easy to read textbook, this is the one you get. Unfortunately, it's out of print, so you have to get it from the library. And a lot of textbooks, I mean, it seems like there's a lot of plant breeders who think we need a good textbook. They saturate the market for 2 years and they go out of print, which is unfortunate. That's why we use our course notes. Okay? Okay. Well, so now let's get into some of the chronology. And what we want really for the first lecture is to give you just a little feel about plant breeding, and at the same time you can start thinking about how to refresh yourself on any genetics that you're not really familiar with, or you might want to become more familiar. And so you want to be doing that between the first and second, maybe third lectures. Okay? Okay, any questions at all? Always feel free to interrupt at any time if you have a question. And always, excuse me, always remind me to repeat the question, because the microphone's so close to me, the archives will get my voice, but they won't get your question. Okay? So some of the important facts, and just to give you a history, because, you know, agriculture's been important for a long time, right? 10,000 years is basically the history, you know, modern history begins about 10,000 years ago. It coincides with the origin of agriculture, and that's probably because people now could develop, you know, written languages, had the time to do those types of things, traditions, whatnot. But actually scientific agriculture is much less than that, and that's things like 650 AD, hand pollination of dates was in practice. That's a way of getting hybrids. Also, the date trees, I believe, have male and female trees, so you need to have different types. Okay. They discovered in 1676 that the stamens, which are the anthers, are the male parts of plants. You know, they isolated female mulberry plants, emasculated castor beans, removed stigmas from corn to improve sexuality among plants. Now that's kind of amazing because remember, corn is a New World plant, so it was not even known in the Old World till 1492. Columbus didn't come back until 1493. So, you know, basically 200 years later, they've started to really manipulate the corn plant for benefits. They produced Fairchild, a new variety, first artificial plant variety, first seed company, 1729. And you can look at these things and see, you know, cell theory developed in 1838. It's kind of amazing. Fella made a lot of hybrids in 1849. Alfalfa came to California for the first time in 1850. So about 160 years ago, alfalfa came to California. Now in eastern U.S. it was called lucerne because that's what the English called it. Alfalfa is because it came up from Chile, and that's what the Spaniards called it. And they called it because of the Moorish influences, where it was a feed for horses. And that's what the Moorish term alfalfa means. So when you think about which crop became more dominant in the U.S., was it lucerne or was it alfalfa? It's alfalfa. And it kept the name showing its origin, which came from Spain through Chile up to California. Mendel, 1866. 1873, Turkey hard red winter wheat was introduced to the Great Plains. That's the wheat that revolutionized agriculture in the Great Plains, made us the breadbasket. Continuity of germplasm, 1881 to 91. Demonstrated fertilization in the fusion of nuclei. First time they were able to figure out how you had fused nuclei to make a zygote. Okay. Great wheat variety called Marquis, which was released in 1914. The cross was made in 1892. Some of the things in corn, ear-to-row method. 1900, everything we knew about Mendel had been forgotten, and they'd rediscovered it. Then you get into a lot of really good things happening in corn, inbreeding in corn, saved ear remnants. Nielsen Ellie did multiple factors in blending resistance, so that was actually in small grains. Hybrid vigor was predicted in 1909. So that's about 101 years ago, right? You know, it's not very long, is it? You know, linkage in corn, heterosis, first mentioned in 1914. Different kinds of hybrids, early 1970 to 1920. X-ray mutations, that was done in 1927. You know, tetraploid maize, doubling, chromosome doubling, which really became popular with colchicine. And then you have things like male sterility in crops in 1950. Now, there's a lot of things that have happened since 1950, but this is where, you know, you get some of the early information on. Okay? Now, for those of you that don't have a textbook and want to print this out, you just click on the print button down here. Okay? Now, I'm going to minimize that. I'm going to stop sharing. I'm going to share, and we're going to go back to my computer screen. And I'm just curious to see if we can pull up the syllabus now. want to be careful about that because it may not show on the archive, because everything's got to show within the window. It's interesting. Boy, that one. Well, let's open. Oh, it doesn't say anything, does it? Well, I'll check it. We'll check to see if the syllabus is there. Okay. Well, in the meantime, we will minimize this. We will stop sharing that. And we will share my computer screen, and we'll go to the next part, which is, what is a plant breeder? Okay. Going to make sure that we got that. Yeah. Okay. And remind me, at the end of the class, we're going to do a roll call inside class. Now let's see. Okay, good. Looks like Susanna, Chris, and Sonia all are all there, can still hear. There's no issues on that. That's excellent. Okay. So what is a plant breeder? Came up? Okay. And by the way, did you see that little window in the bottom, Russell? It always comes across like little X's. Okay, good. That's a pretty good indication. Now, one of the things that will happen is if I ever click on one of these pictures, now this one I can't click on, okay? What will happen is I have to share that picture, so it'll come up and it won't, no one in the archives can see it, so we don't click on those. Some of the pictures are in fact clickable. You know, if you get this and you get the little, you know, the sort of finger on it, you can click on it and bring it up. Okay. The key thing we want to really talk about here is productivity versus production. Okay? Now, I laugh because, you know, I guess if you teach this class long enough, times do change. The whole time, when I first took plant breeding, now that was a few years ago, okay? It was 1973 was my first class in plant breeding, okay? Now, does anybody, first of all, does anybody besides Tom and myself, were you alive in 1973? That's the first question, okay? I mean, you know, that's a long time ago, right? So none of you were. Okay. Does anybody remember anything about history of the 60s and the 70s? About when it comes to agriculture and feeding people. Do you remember anything about that? Anything ring a bell? Were the times there was plenty of food, or was there worry about famines? Okay, and so it was a new generation came out. Okay, Roberto.
There was a famine problem in Ireland.
Well, okay, that was 1860s. That was the potato famine.
Oh, yeah.
Okay, it was 100 years earlier than that. Okay, no, that's fine. That's fine. I mean, it's okay. Okay, in 1966, the Green Revolution started. Have you ever heard of the Green Revolution? Okay, so to put it in perspective, if you ever read the book about Borlaug's life, okay, now Borlaug was the chief scientist that brought the Green Revolution in wheat to the world. Hank Beechel and a number of other people were the ones who brought the semi-dwarf rice to the world. Okay, you will see that somewhere between 20 and 40 million people starved to death in India in the late 1940s. Okay? Now, if you put that in perspective, that's the equivalent of all the soldiers who died in World War II and many of the civilians. Okay? And that was a natural starvation. So in the 60s, you're going to see books like The Population Bomb. It was the Malthusian argument that population is increasing rapidly and food is not. And in fact, we'll stop here for a second because I'm going to show you. We're going to share our whiteboards because I really think the whiteboards are pretty cool. And this is the one reason why I really do like these using this system, okay? So if you do this and you make a graph, I guess I should have made that a little wider. And then if you plot, we'll make it green because agriculture is green. The concept is that production does increase over time, but it's linear. Okay? And the thought was, if you look at food, or population I should say, not food. Food is the green line. Population usually starts out, oh, I don't want to, wait a second, let's not do that. It's not a line. We have to do it this way. It starts out sort of linear, but it actually grows geometrically. And so the idea is that population will eventually outgrow the food. It's a geometric progression. It's a curve, okay? And so then the area that you worry about is in here, this area, okay? What do you do when population is greater than the food that you can have? And the short answer is you starve, okay? Now, so the question that came up in the 60s was, at that time, okay, we were perceived to be right in this area, okay? We were where food and population and production were leveling, were getting to where we were reaching capacity. And so there were famines, fairly large famines, okay? And people predicted that Malthus was finally going to be right. Now, Malthus was the person who in the 1850s suggested that population grows faster than food, therefore we're all going to starve. And it was the Malthusian argument. And so when I took the class in plant breeding, we always had to talk about, you know, are we going to feed the world? Now, does that sound familiar? Okay. So the question is, are we going to feed the world? Well, the short answer is that in the 1960s, pardon me, it just didn't grow linearly. We had a step change. And so we changed the line. And this was due to what was called, and we might as well leave it in green, the Green Revolution. Okay? And that was due to Norm Borlaug and Hank Beechel and their colleagues. Okay? So what happened was we became massively more productive. So for the next 25 years, we heard about, why do you need plant breeders? Because we got all the food. We're paying to store food. We're paying farmers not to grow food. We're doing all these things. So why do you need plant breeders? I mean, we're so, so productive that we're wasting it. Okay? Now, any of you live long enough besides Tom and I to remember those days? Okay, so you probably haven't grown up that way. But that was a big debate. What should we do with agriculture? Now, I've got to tell you some stories. Back in the 70s, with nuclear power coming online, they believed that nuclear power would make energy so cheap that we would have to find ways of basically absorbing it. Almost like, ways of sucking it up, because it's going to be so cheap, we've got to get some way of keeping it semi-profitable. So they did things. How many of you were in soybeans again? How many of you were working in soybeans? Okay. In the early 70s, they told the rhizobial microbiologists that they should destroy the rhizobium collection because fertilizer was going to be so cheap that you'd never have to use rhizobium to fix nitrogen. Okay? That's what they told them in the early 70s, late 60s. Okay? Well, needless to say, what happened is we had an oil embargo, and suddenly energy became extremely expensive. That was in the 70s. And people said, oh my God, save the rhizobium collection, because energy is not going to be so cheap that we'll have to find a way of using it, okay? Wasting it, basically, and the like. Okay. So the Green Revolution made the step change. And what you're seeing now is the Green Revolution looks like it's catching, or population is now catching up to the Green Revolution. So prior to the Green Revolution, we did have surpluses, didn't we? This area under the curve, we had more food than we had people. Now, does that mean that people didn't go to bed hungry? No, of course not. We have enough food to feed people today. It's just we choose not to. Okay? So you should never assume that hunger is based on technology. Hunger is based on the lack of money and the lack of will by those that do have money to feed those who don't. Okay? So you just need to keep that in mind. Okay. A couple things about the Green Revolution. We have some students from India, right? Okay. Santosh is from India. The Green Revolution wheats were sent to Pakistan in 1966, 67, and 68. Do you remember anything about your history with Pakistan in 1967 and 68? Were you friendly?
No. There was a war between India and Pakistan in 1970.
Well, that was 73. There was also one in 67, 68, okay? And they still got the seed from Pakistan to India. So it's pretty amazing. The two countries at war, one has all the seed, and they still were able to shepherd it into India. And that's where the Green Revolution, India and Pakistan, where it made its biggest effect. Okay. Now, the reason why we talk about that is because there's a huge difference between production and productivity. Okay? You should see that little bubble down there. Perfect. Okay. And this is something you always have to... Oh, wait, I went to the wrong one. Pardon me. Should be in what is a plant breeder, shouldn't I? You always have to understand the difference between production and productivity. Okay? Now, production is more than genetics. That's the key thing you have to remember. Production is due to water, fertilizer, pest control, crop variety, tillage, seed condition, that sort of thing. What does the breeder work with of those? The crop variety, right? That's what we change. We create new crop varieties. We don't change water. We don't change fertilizer. We don't change pest control. We don't change the tillage system. And the seed condition is really done by how someone handles the seed once we've given them the genetics. So we work with the variety. So when you look at production, we're part of production, but we're not all of production. Okay? Now, what happens is, if someone comes to you and says, we've got all the food we need, why do we need plant breeders? The argument always has to be on productivity. So I'm going to ask you a question. How many of you have a television in your apartment or home? Okay. How many of you have a radio, say? Maybe that's a little more common. Okay. Well, maybe we should ask this is a young class. How many of you have cell phones? Okay, everybody's got about a cell phone. Okay. Now, going back to the television analogy. In 1940s, 50s, and 60s, all the televisions were built in the U.S. Okay, there were other televisions built elsewhere. Okay, you look at your television. How many of the televisions you have now are built in the U.S.? Anybody have an American-made television? I don't think there is one, actually. Okay, I have a Mitsubishi, which is a Japanese. I have a Sanyo, which I think is Korean, and I have one that's I'm sure made in China. I'm not sure what the brand name is on that, like AMX or something, okay? Now, why did that happen? Is it because we had too much production of televisions? No. Who had the greatest productivity? Somewhere else, right? I mean, that's why manufacturing moves as to where you get it for the cheapest price. So if someone ever says you have too much food, why do we need plant breeders? The answer is you don't, unless you want to have agriculture in your own country. Because it's productivity that says which farmer is going to succeed. So if you think about it, we have lots of debates about genetically modified crops, right? The U.S. has them. Other countries prefer not to have them. How do they protect their farmers from the productivity that's coming out of the U.S.? Tariffs, right? Subsidies, tariffs, things like that. Why do they need them? Because they can't compete without them. Okay? So you always have to know the difference between production and productivity. There's no person I know that wouldn't like, the first color, look at a computer. First computer I bought was about $2,000. It was an Apple IIc. It had, I think, 128 kilobytes of memory and no hard disk. Had to swap floppy disks in and out every time you wanted to store something. Had your program on one floppy disk, load that up, then you put your other floppy disk in and you could take the document, work on it, and take it out. Okay? Now, it cost about $2,000, right? Well, what do you buy for $2,000 in the computer today? You buy, the last computer I bought for my office had a 22-inch screen, has 4 gigabytes of memory, has a 500 gigabyte hard drive, and I don't know how much, and it's $1,000. That's productivity. Okay? So we always work on productivity. Everybody wants to get more for less, and that's what productivity is really all about. Okay? Now, you don't have to worry about, surpluses as much as we used to. So this is sort of your professor being beat over the head and shoulders a lot of time. Okay? You don't have to worry about that. In your case, it's going to be how do we feed the world? How do we create energy for the world? All the other things that's being drawn on agriculture. Okay? New varieties must improve productivity. And they have to keep pace with what's changing. This is a biological system. It's not constant. It's not like you're refining oil. It's not like you're making aluminum from ore. Insects change. Diseases change. There's new cultural practices that happen. One of the reasons why the Green Revolution changed is because they had a wheat plant that had strong enough straw that you could put enough nitrogen on it to really get the yield up. Prior to that time, if you put that much nitrogen on, it would fall over and you couldn't harvest it. Okay? So the cultural practices changed. They added water so you could irrigate, so that your irrigation then allowed you to get much higher yields too. Okay? And you also actually have to predict the future. One of the things you need to be aware of is that when I make a cross, it'll be roughly 10 years. In wheat, it's going to be 12 years before the line is released. Okay? So that's what goes on. Now I'm 59. Okay? I'll be 71 when the crosses I made this year will be released as varieties. I don't intend to work that long. Okay? But, so I have to predict 12 years from now what it's going to be like. So don't think that, plant breeding is, you can make a cross today and have an answer tomorrow, right? You have to have programs that are big enough and broad enough to sort of take whatever comes your way. Make sense? So it's an art and science of changing and improving the heredity of plants. Now, personally, I never like to use the term art, okay? Because a lot of people don't think plant breeding is a real science. And yet, you look at the science that we bring to this field, the statistics, the genomics, the end-use quality assays, the understanding of diseases, the understanding of insects, the understanding of carbon cycles, of nitrogen cycles, all of that. And then you have to overlay on top of that, you have to make a business out of it, okay? It is a real science. That doesn't mean that you can't be intuitive. And that's probably where the art comes from. If you ever think about Einstein's theory of relativity, not that you probably spend a lot of time thinking about it, but do you know how he came up with the concept that light must travel at the speed of light? And when you approach the speed of light, time stands still. Do you know how he figured that? He was on a train. And as the train left the station, he looked at the clock. And he got thinking that if that train could go the speed of light, the clock would stay the exact same time, wouldn't it? It would never change. Sort of kind of like this lecture right now, right? It seems like the clock never changes. I'm just teasing. Okay, but that was his whole point. His point was that if you were traveling at the speed of light, whatever you look back at would be frozen at that time that you left it, right? And so that's where he got into the theory of relativity. Now, how many people do you think got on a train and left the station and never thought about what would happen if the train traveled as fast as light? Now, you could call that art, right? You could call that intuition or intuitiveness. And that's the art of plant breeding. Some people are better at looking at plants and saying, man, this has got what I want. Other people have to rely on statistics and harvesting a lot of plots and things. Okay? So there is a bit of an art. Usually the art is in asking the right question. Okay? Because you think about it, who would have chosen semi-dwarf wheat? And in fact, there were semi-dwarf wheats grown in Italy in the 1920s. But there was no fertilizer, okay, and very little irrigation. So they were just average wheats. It's only when you put fertilizer on and irrigation that these wheats became very highly productive. Same thing's true in soybeans. Sometime when you're working in soybeans, talk to Jim Specht about the first dwarf soybeans. Okay? You know, the soybean breeder at Ohio State, Richard Cooper, decided that you could make soybeans smaller and they ought to have the same effect as the Green Revolution. And he made them smaller. And what did he have to do? He had to change the row spacing so they'd grow tighter, so they'd cover the ground better. And then he found out that they fluctuated. They were just average until he added water, and then their real productivity showed. Okay? So they changed the cropping system almost to make the genetics work. Okay. So what are the types of things plant breeders have to do? They have to recognize the morphological and physiological and pathological response of species that are important. I mean, you can breed for a lot of things that are unimportant. I mean, you know, does a spring wheat breeder need to breed for winter hardiness? I don't think so, because they'll never see a winter, right? Do I need to breed for a, like rice, where you have to breed for the ability to tolerate flooding? No. Wheat's never grown where you flood. Okay, so you can breed for things, but you have to know which ones are important. You have to design techniques that will evaluate the genetic potential for these traits and strains in appropriate species. You have to have a good assay. One of the things we'll discuss is heritability. Heritability is the amount that's controlled by genetics divided by the amount that is the variation in the phenotype. And the phenotype's due to the genetics, the environment, and the G by E. Your assay is part of your environmental variation. If you have a poor assay, it adds experimental error. And so maybe it's not, I should be careful. I should say the phenotype is never measured accurately, so you always have experimental error. If you have a bad assay, your heritability always goes down because you can't measure the phenotype accurately. Okay? So you have to have very good assays. You have to have genetic differences. You have to be able to have new sources of genes. And then you have to be able to combine all of those. So you need a course of action. You have to be aware of your limitations, especially if your assay is poor. You also have to be aware of limitations in genetic material. You know, can we grow wheat at the Arctic Circle? Can we grow corn at the Arctic Circle? No, it doesn't have enough winter hardiness, right? It freezes out. You have to pick the best plant breeding tool, and then you have to monitor the plants. Everything comes back to understanding the biology. Fundamentally at its heart, all geneticists are biologists. Sometimes they may not like that, and sometimes biologists don't like to claim us, but fundamentally at heart, we're all biologists. Okay? Okay. How are we doing so far? Everything's going okay? Now we'll go into the success of plant breeders. And, you know, here you can see all the chapters you can read if you want to. Wait a minute. Why is that called the success of plant breeders? That's, oh man, maybe they don't have the right link. Let me pull that one down. That was my mistake. Let's see if there are two successive plant breeders. That got us into the pedigree breeding method. Let's look at this one. It's breeding in self-pollinated crops. Don't want that. Okay, let's try this one. Now this one, maybe we have both. Okay. I think they put the wrong link in. Okay. Well, in that case, you have to be careful on your feet, you know, when your links aren't working properly. What we'll try to do, if I can work this one out, we're going to tell Terry that we've got a bad link there. Okay.
Is that it? Right there?
Which one?
It says successful.
Wow. Okay, we got the right one then. Come up. Now I'm really confused. I'll have to check that out. I don't know what happened there. Okay. One of the things that's really nice about plant breeding is it's not an abstract science. It's really measurable. You can determine whether you're doing a good job or a bad job. And sometimes it may not be your fault. Don't get me wrong. You know, if you don't have the right environments to select in, if you don't have the right equipment, if you're not properly resourced, if you don't use your resources, now that becomes your fault correctly, you may be at a disadvantage. And I always give the example, and Tom likes this because he's a corn breeder, that there's about 2,100 breeders in the U.S. in the last census of plant breeders, of which 900 are in corn. Okay? So you can either assume that corn is where all the money is, which it is, or two, it just takes one heck of a lot of corn breeders to equal one wheat breeder because there's only 120 of us, you know. So I mean, you can assume that, and there's about the same number of soybean breeders too. So you can assume whichever you are. But what you'll see is that over time, and between 1930 and 2007, if you look at the wheat crop, the state averages, or the national average, went from 14 bushels. Now, I apologize. For those of you that are used to the metric system, we report yields in every kind of unit you can imagine. Okay? For those of you that need to know, 15 bushels per acre is roughly 1 metric ton per hectare if you're using a 60-pound bushel. Bushel weights change. I'll try to convert these if you need them. But this table is hysterical to me because it's in all kinds of units and it just shows you. By the way, for those of you that are a little interested in about American history, America adopted the metric system as its national system for weights and measures in the, I think, 1790 plus. They did that because they didn't like the English, and they didn't, you know, we almost changed the language from English to German. We changed the side of the road your carriages go on. You know, the English always drive on the left side. We drive on the right side. That was to sort of let the English know we weren't going to be like them. And then we took the French measuring system, which is the metric system. Needless to say, the adoption was relatively slow, seeing as you still see miles and bushels and all that stuff. But actually, the metric system is almost, it codates or is synchronous with the Constitution. Okay? Anyway, what you want to look at is the percents because they're unitless. So wheat in 77 years has gone up 280%. Rice has gone up 342%. If you're wondering what CWT is, that's 100 pounds. How many 100 pounds per acre? Corn has gone up 740%. So I guess those, you know, that 9 to 1 or that 9 to, it's probably more like 7 to 1 advantage probably has been helpful. It's also that corn is grown on the better ground. Oats, very small breeding community. It's gone up 189%. Barley, 250%. Do you know why? A lot of money in malting barley, right? They're a much better resource than oats. Sorghum, 690% roughly, not too bad. Cotton, 550. Sugar beets, about 200%. Peanuts, 400%. Soybeans, 300%. And that's probably going faster now because there's a lot more commercial interest in soybeans. A lot more tools are put on it. Potatoes, almost 1,200%. Which actually is remarkable because if you go to the American store, what kind of potatoes do you buy? Russet Burbanks, right? When was the Russet Burbank? Burbank was like 1900. So that's the potato you buy It's 110 years old. The total production is 1,200% better than what it was back then. But you still come back to the same type. I mean, the variation in potatoes is probably among the most in the world. Okay, maybe wheat is close to that. And yet you still get the Burbank potato when you go to our stores. You know, it's that sort of beige skin potato, the baking potato. Okay, it's just amazing. Alfalfa 171%. Okay. Now there's an equation here that you have to know. Okay, and if I wake you up in the middle of the night from the deepest sleep, and I ask you what makes the phenotype, you're going to be able to say at the end of this lecture, it's the genotype, the environment, and the genotype by environmental interaction, right? That's the fundamental equation. Now, the phenotype is what you see. It's what you measure. The genotype is the genetics that control the plant's genetics, okay? And how it grows, develops in that. The environment is what that plant grows in, okay? And the genotype by environmental interaction is how they interact with each other, okay? So now, let's think a little bit about that, okay? If I grow a plant in the dark, can you tell the difference between an albino plant, now that's one that has no chlorophyll, and one that does have chlorophyll, that's grown in the dark? No, right? Chlorophyll needs light to go from pro-chlorophyll to chlorophyll. So it's a white plant as long as it's grown in the dark. Okay, if you grow a plant without diseases, in the absence of the pathogen, can you tell if it's resistant or susceptible to the disease? No. That's again a genotype by environmental type of interaction, right? Okay, if you grow plants that under irrigation are massively different without water so that they're under drought, will you see the differences that you'd expect to see under irrigation? No. That's all different types of genotype by environmental interaction. Okay, now, let me give you one other comment that you need to know. How many of you are taking statistics? Okay, how many of you have ever taken, statistics? Okay. One of the questions, have they ever asked you about interactions? So like, that you have a cultivar by an environment interaction, or you have a main effect by like a year by location interaction or something like that. Have you ever seen those? The number one thing you need to remember is you always have to have two of whatever you're having to have an interaction. Okay? You can't measure an interaction if you only have one genotype. So the interaction is if the genotypes respond differently to the environments. Okay? If you have only one environment, you can't tell if they're responding differently. I mean, you might, well, I guess it's careful. I mean, you can't say there's an interaction there. You can say that they grow differently in the same environment. But whenever you do an interaction, it always has to be two cultivars. That's the first thing. So if someone says, I'm growing cultivar A, and I'm growing it in one environment, do you know if it's a G by E, a genotype, or an environmental interaction that's affecting that plant? Not at all. Okay? So I'm going to, for a second, just stop here, because I want to basically what you want to look at is if you have a graph and you have two cultivars, right? There are different kinds of interactions. That's the first kind, and we'll talk about that's a change in magnitude, okay? There's a second kind, just like this. That's a reversal in order. So the first one is a change in well, tell you what, let's move that over to here. Now, one thing you'll notice on this, Russell, is whenever you do this, you always have to extend beyond it and go further down. And the reason for that is that the second line is often, if it's the last line, it's often not seen. Okay, so it's known as a change in magnitude, which means that the lines differ because they, one becomes progressively better. As a breeder, that doesn't bother you, okay? The red line's always better than the purple line, right? So that doesn't hurt you at all. If you're going to make a recommendation to a grower, grow the red one. Everywhere in that curve, it's still the better line, right? The change in order or reversal in order is the one that concerns you, because the blue line is better in the lower part of the x-axis, but worse in the higher part of the x-axis. So how do you make a recommendation? That's the problem, okay? So you have to be very, very careful when you're doing these kinds of things, okay? Now, if there is no G by E interaction, what you can see is something like this. I have to be a little careful. For those of you that have to teach at some time, you never want to use blue and green. Do you know why? Colorblind. What's the most common colorblind that you have is blue and green. Okay? So you never know if someone in your class, I mean, quite frankly, among men it's more common than women because it's a sex-linked trait. And so it's quite common that blue and green will look almost identical to a person that's colorblind. They can't differentiate them. I actually had a technician who worked for me, a really good guy who was colorblind. It was really amazing because he could not tell when the wheat went from green to beige. He could not tell when it was really harvested. So what we'd have to do is we'd go out to the field and you just thresh it out. If it's still, you know, green, it was okay. I mean, it'd be milky. Otherwise, you know, he had to do that. And it was interesting because one time he went in and he, with the combine, and he cut the border, and the border was a very late maturing wheat, so he said it was too early to cut. Well, the main field was all earlier than the border, so that field actually could be cut. And so you always have to worry when you're teaching about colorblind. Okay, enough of that story. If there's no, whoops, that's not what I meant to do. If there's no interaction, then what you end up with is something like this. I'm going to see if I can make this work. Does that look parallel? Okay. When they're parallel, there's no interaction. They're responding to the environment in the same way. It still happens in this case that sort of deep purple line is always better than the light purple line, but there's no interaction. The response is identical. Okay? So you can have lines where there is no G by E. You can have lines where there is G by E, genotype by environmental interactions. And basically in this case, this would be the environment right here. And up here is whatever you measure, you know, your trait. Okay? So you can have it where like the red line is always better than the purple line. That makes it easy. It's only when you have, now the other name I should mention for this, reversal in order, the common name for that is also a crossover interaction. Okay? Because the lines cross. So it's known as a crossover interaction. That's the one that causes you the most trouble when you're trying to make recommendations to people. Okay? Okay. So we'll stop sharing this. And now we'll go back in and we'll share. Oh boy, now I probably shouldn't have done this because now I've got to figure out which one is the success of plant breeders, right? Well, we got that one lucky. Okay. So how do you determine if you've made improvements? You know, you can't do it on production, right? Because if it's just production, it could be that you've got fertilizer, okay? It could be that you're irrigating. It could be that now you've got weed control or fungicides, you know, disease control, right? So you can't just do it on production. And basically, you have to do it in some way that looks at least two cultivars, right? Because you're looking at an interaction. You know, you're looking at the productivity. If you look at one cultivar, you can't tell if it's better than a second one, right? Because you're not looking at the second one. So you can make comparisons of new lines to historical checks. That's one way of doing it in things like regional nurseries. Now, the reason why that works is because the nurseries are in the same environments. So if you're better than the historical check, the older line, and it's in the same environment, it's not due to the environment, right? It could be due to the genotype by environmental interaction, but that's and it probably actually is, and we'll show you some things. But at the same time, it's also due to the genotype. You can do very designed experiments, which include things such as historical checks and cultural practices, where you now can compare the cultural practices and the historical checks and the modern cultivars and the modern practices all at the same time. The best work that's done on this is actually by Pioneer. And they've got a wonderful deal where they show corn hybrids from like the 1920s up to the current day. They look at it under the cultural practices that were done in the 1920s and the cultural practices that were done today. And they look at hybrids that were released in each decade in between. And they have these really beautiful graphs, okay? Now, each of these will have a little bit of confounding, okay? Which means that you can't separate truly the environment from the genotype, but you get a much better picture of what's going on. Now, I'm going to see if I can bring up the okay, not the syllabus here. Okay, we want to go to external links, and you're going to have to tell me on this one, Russell, if it's here. Additional reference material that might help you in the class, that's all these books. Graphs. Didn't come up. Okay. So what we have to do now is I have to minimize this, I think. Stop sharing. And now I share my computer screen. And it's probably one of these two. That come up? Yep. Okay. Still there? Okay, perfect. What you see here is a graph. Sorghum grain yields in Nebraska from 1850 to 1880, showing an increase of 86 kilograms per year. Okay, now that's just production. Okay, it doesn't say anything about the variety. It could be the same hybrid for all you know. Okay, and then they looked at what happened. They changed the height. They went to hybrids. They went to green bugs. And so now you can say, okay, the production went up. Maybe it's due to genetics. But again, they haven't really proved it, have they? There's no comparison. It's just the total production. This is where you start looking at, where you start seeing things. Maize grain yields, each point represents the mean of a year. This is their coefficient. Again, this is just production by year. It goes up like this. This is where you start seeing real studies. This is done by Don Duvik, who is one of the great corn breeders, managers, I guess, Tom, is what you'd say. He's probably best known. He was the vice president of research or director of research for many, many years at Pioneer. What you have here is you have three different densities: 30,000 plants per acre, 47,000 plants per acre, 64,000 plants per acre. Okay? Per hectare, is that per hectare? Yeah, should be per hectare. Yeah, that would make sense. And now a hectare is 2.2 acres, so that's why it's quite a bit higher. In the 1930s, you actually grew hybrids that were used to very low density. Okay? So in the 1930s, you would look at these lines, and you can see that the tons per hectare if you release the line, now what they've done here is the regression of grain yield on density on the year of hybrid release. So at this end, this is when the hybrids were released in 1930. This is when the hybrids are released in 1980. And what you can see is at 30,000 plants per hectare, you have a pretty good yield, and it goes up slightly as you go to higher densities. Or pardon me, it goes up slightly as you go to more modern hybrids. Now, if you go to the 47,000 plants per hectare, which is close to the commercial rate of what, probably 10 years ago? 15 years ago, you can see that the 1930s can't take the competition. That's why they're so much lower, okay? But as you now get more modern cultivars, their yield increase is much higher, isn't it? So you're looking at say 4.8 tons per hectare, and a maximum of a little over 7 tons per hectare with modern cultivars. If you put them at high density, now they're down to 4 tons. So if you just looked at densities, you can almost say that the higher densities are really a detriment to the earlier hybrids, right? I mean, they lose yield at those high densities. And if you go to the 64,000, they become, they're equally poor. But they actually, as you come up, you can see even the older hybrids tend to, or as you become more modern, they're still lower than the 47,000. And it's only when you get to the very end is the high density really become beneficial. So in this case, you're separating out the environment, which is the density, right? Now these are all tested at the same environments. So you've got the environment's held constant, the density held constant, or changing, I should say, 3 densities, and the hybrid changing. So this is a very good way of looking at, do you have genetic improvement, okay? Because at 30,000 plants per hectare, the old cultivars yielded 5 tons, and at the modern cultivars yielded basically 7 tons. Now that's the same density, same environments, right? So the only thing you're differing now is the genetics. But the reason why Don did this is he said, is it fair to compare new cultivars versus old cultivars at the current production practices, which would be at 64,000 plants per hectare, or maybe that's a little high, which he's getting now virtually 9.5 tons versus 4 tons, okay? So that's now the same environment, okay, the same density, but you can see how the age of release responds differently. And what it's really telling you is that at the original densities, the densities that they used in 1930, the modern cultivars are maybe, 1.8 times better? If you go to a modern density, the old cultivars are 2.5 times poorer. So there's an interaction between the environment and the genotype, and if you're really going to see what kind of progress you're going to make, you either have to confound with either the modern growing conditions or the old growing conditions, or test them at both. And then you can really see the true differences. Okay. Now we're going to end pretty quickly here. Again, here's more on the densities. Root lodged, another trait they used, because you don't have to use yield as your estimate. And again, you can see at the higher densities, you had a lot more percent plants not root lodged. Okay, so it's like a negative. Fewer plants, or 60% were root lodged, or not root lodged. 40% were not root lodged, but when you come up here, almost 95% were not root lodged. So they've really made some changes due to density and genotype in that trait. And in fact, let's stop. We'll come back and do the Southern Regional Performance Nursery the next time. We'll show you how you can use uniform nurseries. Okay? So everything went okay? Okay. Well, we'll see you all next Thursday. And if you want to read ahead, you can see the impact of what one program's done. Happens to be mine. I keep better records on mine than a lot of people do. And then you can start reading about the pedigree breeding method. So by the end of next lecture, you will be a bona fide self-pollinated crop breeder ready to explain self-pollinated crop breeding methods, at least one, to your parents, to your significant others, and anybody else you want. Okay? Thank you now.